So this is intended to be a bit a pro and con session, and I have con opinions on digitalization and machine learning, but I will really bring a pro perspective here. So the goal is that I will talk about why do we actually need digitalization and machine learning in infectious diseases and in clinical microbiology. And this, to actually give you the answer right away, this has to do with the increasing complexity of the data we are facing and we are analyzing, and also with the changing interface of clinics and laboratory, the way we communicate with clinical partners. And so in the second part of the talk, I will talk about what are the opportunities, so what is already here. Here I mainly take the point of view from a clinical diagnostic perspective, and what can we do to actually move forward. So Jacques Schrenzel already said it in the beginning that the field of microbiology or clinical microbiology has tremendously changed over the last 2 centuries. So nowadays we are here, we have very fancy microscopes, we have total lab automation. The way we identify pathogens has radically changed, basically from high costs and biochemical reaction profiles which take ages to seconds and very low cost identification, and even more so in the field of molecular diagnostics with the introduction of single cartridges and multiplex PCRs, and now the sequencing technologies moving away from short read sequencing to ultra fast long read sequencing technologies. So a lot is going on in the field of clinical microbiology. So we can break it down into 2 aspects in a modern biological lab. So on top here you have the phenotypic way to identify. So you take a specimen, you culture it, and in red I highlighted where most changes happen. So I mentioned automation. This also came along with a strong standardization, and this is perfect if you want to use data and make data available. You need standardized data. Then automated image analysis, which I will also later on talk about. Then the identification process here, faster identification, lower costs, and a very high resolution. And I personally still think that MALDI-TOF is a very underused resource. There's so much information in a profile, and we simply use 10 peaks to identify a pathogen, and much, much more can be done with this information. And I think this will also happen during the next couple of years. And then we have the genotypic way here, so DNA and RNA, where we measure basically multiple genes, we can sequence them. We can also include resistance testing here. A good example is Mycobacterium tuberculosis and rifampicin resistance detection. And now this moves to panel PCR, which is basically the preparation for metagenomics, which will also sooner or later come to clinical microbiology. Now, there are 2 examples here in particular of the genomic field which highlight the amount of complexity we have to deal with. So panel assays, when they started a couple of years ago, we have 20 pathogens, antimicrobial resistance, and it really changed the way we do diagnostics. It's no longer a single pathogen we think about, but a syndrome actually comes into play, and we do a shotgun search basically, still a bit selected, 220 pathogens, but newest cartridges now have 30 and more targets in it. And this is a challenge to transfer this information to the physician. So obviously, how do you visualize infinite amount of information, a table at the moment, but if you have 40, 50 pathogens you need to report, how do you best do that? This is a challenge in the communication and also in the visualization of a lab result. And then also costs. So these panels are quite costly, and I think it's absolutely important to measure the impact for the patient as well. And so this gets even more complex if we think about a metagenomic approach where we sequence basically everything in a sample, and this generates a field of a hypothesis-free medicine. So you just take a look what is there, and then you also have new concepts which come into play. So for example, what is a pathogen? If you detect the signal, and you actually then see maybe relationships of other bacteria around that pathogen, and I think this gets more and more complex. Again here, what is the true impact for the patient, and how do you visualize a metagenomic data set that somebody who, a clinician who just has a couple of seconds time to actually grab the most important information, how do you transfer this information? So I clearly think these are challenges which will further increase. So there is, due to this technological development, an explosion of information ongoing. So first of all, there's an increasing demand of healthcare data for personalized medicine, for value-based medicine, for cost control. And due to the technological capacities and possibilities, the amount of stored data on the planet, and you see here, I mean, this is an exponential growth here of available data around the globe, and it stopped here in 2010. And you can see here, these are exabytes of data, and 1 exabyte is 1,000 petabytes. So it's ridiculous how much data we actually produce around the globe, and the same is true in the lab. So there's a trend to have this rapid increase, and the doubling time of knowledge is becoming shorter and shorter. So this is the life science data half-life, basically 73 days. Now imagine you go to med school nowadays, it's a nightmare because you don't know what you actually learn. I mean, two months later, it's already outdated. And I think this is really a challenge. How will we get information? How will we get the right information to treat patients? So, and besides the amount of data, the data is also complex and interconnected, as we heard. So clearly some challenges here ahead. And due to this explosion and complex information, there's an absolute need to better structure all this data and to have new tools available which help us to find the way through this jungle here and really generate a meaningful impact for the patient. So we will need tools to manage and interpret this data, to also communicate this data, to generate a clinical impact for the patient, and also communicate to changing stakeholders. So for example, patients, they also inform themselves more and more. They will have such tools and information available. As we heard, data sharing of your own data, so you maybe will analyze in the future your own data, and then you confront a doctor all of a sudden with a machine learning algorithm, and what do you say then? I mean, this is clearly important that we get familiar with these kind of tools. Now, to basically define where to start, I thought about the diagnostic process of, this is actually a turnaround, the diagnostic process. So you have the clinical side where you have a patient. In this case, it's an influenza-like illness, a patient with cough, fever, and an acute illness. And you send a sample to the lab, and this is called pre-analytical process. And here, for example, a machine learning algorithm could help you to identify if the correct swab was sent or what was the quality of the sample. So a pre-analytic quality control. It could also help you to use the concept of a digital twin. So this is like I imagine, like a shopping list when you go and you say other customers also asked for this and this product. So it could be that, for example, in such an algorithm makes suggestions of what else could be useful. Now, in a let's say, otherwise healthy person, maybe the suggestion of the digital twin would tell you don't order anything else. I mean, this patient has an influenza-like illness, but if this is an immunosuppressed patient, maybe with a leukemia, the algorithm would suggest you to order a more broad panel diagnostics. Then in the lab itself, you have the analytical process, and here as well, machine learning algorithm can help you to run some quality controls. I mean, at the moment, you do this manually. For example, if your test indicates that there's a flue A and B at the same time, this is very unlikely, so most likely the cartridge has an error, and then perhaps such an algorithm would right away reorder a test so that you repeat it, maybe even fill out the material vigilance form for you so that you do not need to do that. Then an expert system might help you to support, for example, automated reporting. So it could send messages out to public health and provide data. It could also be used locally, for example, for outbreak detection. And then on a post-analytical level, and this is the communication back of the data to the clinician, it might help you to visualize information, for example, generating an epicurve, highlight important data. So, for example, if this is if you have genomic information on this virus available, it could be that it detects a particular virulent substrain, and also clinical decision support could be provided. So maybe the algorithm in the background runs a risk analysis of this particular patient type and tells you if it's better to actually hospitalize such a patient. So a lot of opportunities basically where machine learning may have an impact, and you can translate that to any other field, for example, blood culture diagnostics or sepsis and many other things. So the next revolution, data-driven microbiology. So is this a world of opportunity? I think what is required, and we heard about that in the previous talks, we need interconnected and interoperable microbiological data. The data is required to truly understand the diagnostic process in a systems approach. So it's not only as I highlighted in the slide before, your own lab, which you can pretty well control and understand where the data comes from, but also to realize that you're part of a whole system, and the pre-analytical and post-analytical steps are as important. And this may offer then improvements in the quality due to standardization and interoperability to monitor the diagnostic process. So for example, you will realize where are particular bottlenecks, where do you perform very well, and this is also, these are good showcases, I would say, to finance such a system. We heard about that in incentives basically to invest in structured data could be well due to business intelligent arguments. Then optimization of the diagnostic process, support in interpretation, I mentioned expert systems, simulation of changes in the process. So what happens, for example, if the emergency room is completely filled up and nobody's, because they have so much to do that samples are no longer coming to the lab. So you could really also simulate certain scenarios. Then digital biomarkers, I will also talk about that a bit, and then surveillance of changing data. So I have brought a few examples, and we will certainly hear more about this particular example, and Chuck already showed it this morning because Ken Smith is also is here, I think, already, or maybe he will talk tomorrow for sure on this. And this is a wonderful study on actually a digital microscope, or a microscope which generates pictures and scans. And you can see here a blood culture smear, very messy, but a few bacteria in here, and here in the magnifications, you can see, for example, the bacterial cells. And in a training data set of, and I'm surprised, of only 180 slides, and this is something you generate in a few weeks, I would say, in a lab. So they generated a lot of these snapshots, let's say, of bacteria. And then they trained an algorithm. You can see here the learning elements and the performance overall in the training data set. And then they took a second independent data set to check for sensitivity and specificity, and it was highlighted already in this morning. Gram-negative bacteria, for example, could be correctly identified with a 98% sensitivity and 96% specificity, which is excellent if you think about how long lab technicians also need to train, and so this is a really nice performing algorithm. Obviously also some issues here, 75% sensitivity, but I'm in the pro session here and not in the con session. So, but for sure, I mean, this is something maybe a better trained algorithm could also compensate for. So opportunities in microscopy, there are many. The concept is clear, it's pattern recognition, and this concept can be used for many, many things. So it could be a plain detection of bacteria, fungi, parasites, and leukocyte reactions, let's say in a biopsy or in a swab. It can be very qualitative. Yes, no, there's bacteria in or not in. It could be quantitative. Maybe the bacterial cells and leukocytes could be counted in a field of view and then provide you additional information. Such a system could be trained for many different staining technologies in the lab, gram staining, tuberculosis staining. Maybe it can also be combined with antibodies, with fluorescent tags, so to be even more sensitive. I think this really has plenty of opportunities. The advantages of such a system are clear. High standardized reading versus a manual analysis. So in our diagnostic lab, we often do a microscopy competition between the lab technicians. So they all look at the same slide, and then we check who is best, and you know, the person then wins basically. And you see that there are, dependent on experience, differences, and a machine learning algorithm obviously is never in a bad mood, is never tired, so performs always the same way. And so it could also be run overnight, certain samples which do not need an immediate workup, so you could work them overnight, and you would in certain ways also saving technician costs because you could use them for different purposes. Studies, there are not many studies published so far. I personally am only aware of this particular study, but perhaps there were more recent ones. But this is certainly something where this whole field will now start to also generate more data here. Then other aspects here in terms of pre-analytics, maybe the sample quality could be evaluated. So if it's contaminated, for example, with cellular debris or epithelial cells. In analytics, there are a series of advantages. So to monitor the turnaround time for critical samples, you could really measure that because you have timestamps, so you know when does a sample come in and when is a microscopy result released. You could optimize for sensitivity and specificity, and I would like to highlight this point a bit more. So for example, in a positive blood culture, this is an enriched material, and you know the blood culture became positive in your machine. So the sensitivity is not necessarily an issue because you know the blood culture is positive. And if, for example, your algorithm would not find any bacteria, you would then go and really check them manually. But in non-enriched material, non-pre-cultured material, for example, from a primary sterile site, like CSF or a blood smear or so, sensitivity will be absolutely key, and you would train your system to be maximum sensitive. And then you will also have issues with sometimes colonizing bacteria, for example, sputum samples, or if you check stool for parasites, then certainly an algorithm would need to be optimized for sensitivity and specificity. Then the second example here is digital plate reading, detection and identification, and here a lot is going on. We also heard it this morning, detection of colonies, to just say what is a positive and a negative plate. I think this could beautifully work for a screening of for example, multi-drug resistant bacteria. So there are some numbers of older studies, BD-Kistra system, for example, with very high sensitivity and specificities. Other studies have shown also similar high sensitivity levels. It's also possible to identify bacterial species, not only to say, yes, there's grow or no grow on a let's say, selective agar plate, but to really identify if this is a Staph aureus, an Enterococcus, and so on, or E. coli can be reliably identified with these kind of algorithms where you consider the morphology, the color, the growth rate, and so on. here, just to visualize that with another software, and I think this is also ongoing work, so these systems will become better and better, and I think a typical use case would just be to, at the start, I would say, to really identify culture negative plates when nothing is on it, so that the lab technician does not need to further look into them, and that they can just be released, and the algorithm basically says there's no growth on it, and then it's validated. Here again, the concept is a colony growth pattern recognition. The opportunities here are, again, it could be just a plain growth on a plate. It could be identification. It could also be combined later on, and I think companies are working on that to identify a pathogen and then, based on a machine learning algorithm, use a colony picker and a multitoph-based identification. The advantages here are more or less the same as before, but here I'm also not aware of studies looking into at least these processes here in more detail. From the analytical point of view, a series of benefits, I guess. Detection of bacteria on an agar plate. So I think this is really an interesting tool to identify multi-drug resistant pathogens, and it can be used to identify, as I mentioned, based on these various aspects, and perhaps also e-tests and Kirby-Bauer disk could be read, so that would be extremely helpful in the lab. So post-analytical impact, clinical decision-making. I think this is an interesting topic, which we will also further discuss in this, and we just heard about clinical decision-making in empiric antibiotic usage. So there's clearly the question, what actually changes the clinical decision? So adding, for example, an indication process for a therapy and an intervention. For example, modifying the therapy, choice of drug, the dosage, the duration. Could an algorithm suggest you this? So, for example, if you realize there's a colonization with Streptococcus agalactiae in a pregnant woman, would then the algorithm remind you to use, for example, an antibiotic prophylaxis before delivery? That could really improve the quality of healthcare. And then obviously also the impact for the patient in determining reducing morbidity and mortality. I think this will be pretty tough to to assess and to measure, because these are hard outcomes, and just to have measuring basically the impact of a diagnostic test with such outcomes will require large studies and also multi-center studies. So here I was thinking about the threshold model, and this is actually an idea I got from a colleague from Tanzania, where he showed the probability of diagnosis. So you have a certain threshold to diagnose something, and this is the threshold A here, and above this threshold it's actually worth to do a test. And then you have a treatment threshold, which is B, and if it's below, above this threshold, then you would anyways treat. So your diagnostic window is between A and B. So it's worth to do a test because there's a therapeutic consequence. So the question is, what is the impact of a digital biomarker on this model? And I think what will happen with digital biomarkers, this diagnostic window will most likely get wider in the ideal situation, because you save some time. You will recognize a disease earlier, and perhaps due to the differentiation, and we heard this from Christopher Seymour's talk, that you can really, based on a better understanding of the data set, you can give various sets of treatments to a single patient, and not longer just option B. You have other options available. I will, for the interest of time, skip this example. So this is a very famous digital biomarker study, which you probably all know. I just want to spend some time on the communication, which has to adapt. At the moment, we are producing in the lab simple single results, I would say. It's lists, short lists and categories of results. And this is very much focused on a single result from a single time point. And the clinicians, on the other hand, they have algorithms which often are binary, and so it's a left or right, black and white decision in an algorithmic tree. So they're also dependent on these types of results. And what will happen with more complex results, interconnected data, all of a sudden we have series of time points we analyze. We probably also have a more profound understanding what is a pathogen. The role here will be much more complicated to interpret. And I think this needs a discussion board, maybe something like a data integration board where microbiologists and infectious disease people meet and discuss the results and have various visualization tools available. So where should companies invest in this field? I think one thing which I was missing so far in the discussions are the prospective validation studies. Really, most of the data is analyzed retrospectively, but prospective trials are needed. Then excuse me. Then I think data integration tools for the laboratory, diagnostic stewardship tools could be very interesting, and I highlighted this example. Identification of species, which I also shown you, telebacteriology basically. And then antibiotic resistance, so here expert systems which help us to analyze the data. Hospital epidemiology surveillance, I think this could be extremely interesting if we have the identification integrated in a certain way that, for example, if you find XYZ amount of Serratia mascescens, you realize, oh, there could be an outbreak if this is linked to a ward, and that there are automatic warning systems. And then public health surveillance, obviously, and something which I really like as a topic, this Internet of Bodies, which I think is a nice term, that wearables and out-of-the-hospital data is more and more available, and this could be an extremely interesting future field. So the take-home message. I think we are still here we still have these two axes, the phenotypic world and the genotypic world. The question is, what are the drivers here for machine learning? And I think it's the technological development, the requirements of patients to have a most personalized approach for themselves, then valued-based medicine, which I did not explored so much, but I think it's clear that we need a certain cost control, and I think here data-driven healthcare, but also data-driven microbiology really has an impact on the patient. So with this, I would like to thank all my team. It's fantastic to work with them, and would not possible to sell you here the car, the Porsche. And I really think it's wonderful. And we have a lot of data incentives, also a few posters outside on this topic. And with this, I would like to thank you for your attention.